It's a very great pleasure for me to welcome to the podium Professor David Vaux, whose work, of course, initiated the journey and who is now Deputy Director of WEHI. David.
Suzanne mentioned Andreas's chair, and just to add, I gave Andreas that chair, but it was a very special chair because it was given to me by Ishwar Hariharan, and it was given to him by Alan Cowman. So I hope that one day we'll have a nice Perspex cabinet, and that chair will be put in that cabinet and will sit down below in the Galleria where it belongs. So this is a slide illustrating publications on cell death. And in actual fact, the first publications on cell death were way back in 1842. But the modern era began with John Kerr, also an Australian, who came up with the term apoptosis in 1972. But he'd actually been working on cell death earlier than that, back in 1964, I think his first paper was on cell death, and then the term programmed cell death was coined by Richard Lockshin, and then apoptosis, the technical term for cell death, by Kerr, Wiley and Curry in 1972. But you can see that the number of publications per year was extremely low. Hardly anybody was interested in this process of cell death or apoptosis. Even the developmental biologists such as Bob Horvitz, who were studying cell death in the worm C. elegans, that generated relatively little interest. But come the late 1980s, suddenly things changed and the number of publications on apoptosis grew exponentially. And now there's something like 25,000 publications, new publications appearing every year that mention the term apoptosis or programmed cell death. So this talk is about what happened to cause that change in interest. So I'll take you back to 1987, so 30 years ago, and those times were very different. At the time, there was a President of the United States who nobody ever expected would be a President of the United States. So it'll probably be very hard of you to imagine how things were back then. And this is some of the people in Jerry and Suzanne's unit back then. You'll see there's me looking very young. You can see some of the hairstyles. The perm was in. That's Liz Webb, who generated many of the transgenic constructs. Here's Aura Bernard, who cloned the mouse homologue of the MYC gene. There's Ishwar Hariharan, who gave the chair to me, then the chair I passed on to Andreas Strasser. There's Alan Harris, and here's Helen Rosenberg, who was the first person to generate a transgenic mouse in the unit here at WEHI. So that's 1987, and don't we all look young? Now, the story of BCL2 really begins about 10 years before that, and that's through the work of Janet Rowley. Janet Rowley was a haematologist in the United States, and she was studying the chromosomes in a variety of lymphomas or cancers of the blood cells, and she noticed that in almost every case of follicular lymphoma, there was the same translocation. Now, we all get two chromosomes of each type. We get one from our mother and one from our father, and you can see here that in this chromosome spread, which comes from a patient, and it's obviously a woman, you can see the two X chromosomes. She was a woman with follicular lymphoma. You can see that there's an abnormal chromosome 18 and an abnormal chromosome 14. Now, of course, I never would have picked up anything like that, but Janet Rowley, because she looked at so many of these things and she was so sharp, she noticed that a little bit of chromosome 18 had broken off and become stuck to the end of one of the chromosome 14s, and vice versa. So this particular chromosome translocation is seen in practically every case of follicular lymphoma, and that suggested that maybe there was a gene at the breakpoint that was responsible for turning a normal cell into a cancer cell. And that gene was called BCL2. It was named BCL2 by Carlo Croce, and he was the head of one of the labs, and it was in his lab that Yoshida Sugimoto was the first person to clone the BCL2 gene, which normally resides on the end of chromosome 18, but is translocated into the immunoglobulin locus on chromosome 14 in cases of follicular lymphoma. So that was like a smoking gun. A lot of labs tried to clone BCL2, and Sugimoto was the first person to do so, but another person who did so independently was Mike Cleary at Stanford. Now, Jerry and Suzanne were very aware of the importance of translocations in cancer because, of course, they were the ones who generated the transgenic mouse that recapitulated a different chromosome translocation that was seen in a different kind of lymphoma, namely Burkitt lymphoma. And this is a letter here written by Mike Cleary in response to a request from Jerry asking for the BCL2 cDNA. And in this letter, Cleary very kindly offers to send the BCL2 cDNA, and he said you can more or less do whatever you want with it, but don't make transgenic mice, because that was what Cleary was intending to do. So Jerry gave me the BCL2 cDNA. I'd been in the lab a couple of years and had been interested in growth factors, but he gave me this BCL2 cDNA and said, well, can you figure out whether it's a cancer gene or not? So back in 1987, as Suzanne said, there were a number of oncogenes or cancer genes known, and the thought was that these cancer genes are really, their real role is to regulate cell growth and proliferation. But when a cell turns into a cancer cell, it's because these cancer genes or oncogenes are either mutated so that they're always in the on state, or their regulation is abnormal so that they stimulate cells to grow and grow and grow and proliferate. One of these genes in particular is called ABL, and there's a viral counterpart called VABL, and it was also being studied by researchers here at WEHI. And in particular, I'd like to mention the work that Wendy Cook carried out, because Wendy Cook was studying VABL, and she showed that if she put it into a cell line called FDCP1 cells, and this is a cell line that was generated by Don Metcalf many years before, this cell line requires a growth factor to grow, and if you add the growth factor, it grows like a weed, and if you take away the growth factor, the cells die. And if you take these cells and inject them into a mouse, the mice don't get tumours. But what Wendy showed was that if she put into these growth factor dependent cells the VABL gene, the viral activated version of the ABL oncogene, then these FDCP1 cells became growth factor independent, and they would grow and form colonies even if you hadn't added growth factor, and they would form tumours in mice. So I thought, well, these FDCP1 cells seem like the ideal cells to test whether BCL2 has similar properties and will stimulate cells to grow without growth factor and form tumours in mice. But how do you put BCL2 into FDCP1 cells? And for that, again, I was extraordinarily lucky. Here's a picture of some of these cells. These are IL-3 dependent cells in the presence of IL-3, and these cells are all round and healthy. These green ones and red ones are the ones undergoing apoptosis. And if you take away interleukin-3, within a day or two, the cells commit suicide. They activate an inbuilt self-destruct mechanism. So I thought, well, how do I put BCL2 into these cells? And so I was extraordinarily lucky, because in the bench opposite me, and the provider of the wonderful golden chair of Andreas Strasser, was Ishwar Hariharan. And he developed a retroviral vector, and this retroviral vector allowed you to put genes such as BCL2 and c-Myc into the viral vector, and then you could use that to infect and stably transfer the gene and turn it on in whatever cell type you liked. So I took this virus and expressing BCL2, and I put it into a whole bunch of FDCP1 cells, and I washed them and cultured them with factor and without growth factor. I also infected them with an ABL virus, as did Wendy Cook, to see if there's a positive control, and they all produced nice colonies. But unfortunately, BCL2 seemed to do nothing. And this was very frustrating, because it was very hard to tell which cells had been transfected, because at that stage we didn't have any antibodies to BCL2. The only way to know if the BCL2 virus was working was and transferring the gene was by doing northern blots and southern blots, and because we didn't know what the phenotype was, it was hard to tell what was going on. But I kept looking harder and harder, trying to see maybe even little baby colonies of cells. I never did, but I would see in the BCL2 transfected cells that whereas the normal cells had died in the absence of growth factor, in the soft agar cultures, the ones that had BCL2 were just sitting there. They weren't forming colonies, but the cells were still refractile, they were still alive. And even more significantly, if I took those cells and added back growth factor, then the cells started to grow once again. Now, this seemed very surprising because it was different to the action of other oncogenes known at the time. And to check that this was the case, I isolated many independent clones of cells that each carried this BCL2 virus or a control virus. And so here's some of my notes from the time. So this is July 1987, and each colony, so there's 18 colonies of each type, each is an independent clone of cells, and I've cultured the cells without interleukin-3 or growth factor for 4 days. And these are the control clones, and you can see that V is for viable, and there's no viable cells whatsoever, and D is for dead, and these are dead cells that were present in each of the cultures. Now, I haven't shown you the effect of adding interleukin-3 to the cultures, but if you added interleukin-3, then there were many hundreds or thousands of cells in each well. But here's the cells expressing BCL2 or infected with the BCL2 virus, in which I've taken away growth factor. And you can see here, although there's some cells that are dead in most of the wells, in many cases, and almost every case, there's also some viable looking cells. So this was 18 independent clones in which the cells were still able to grow. And more significantly, if I added back interleukin-3 to these wells, then the cells were able to proliferate once again. So this meant that BCL2 was having an effect. So that made me extraordinarily happy, but the effect was different to other cancer genes known at the time because BCL2 wasn't stimulating cell proliferation. It was stopping cells from being able to self-destruct. This shows a picture from the B-cell neoplasia meeting in Basel in March 1999, where I got to present, and you can see in the same session, here's Carlo Croce and here's Stan Korsmeyer, two of the other early researchers in the BCL2 field. And here's one of the early experiments looking at the effect of BCL2. So one of the thoughts was maybe BCL2 was having a similar effect to just low levels of growth factor. But in this titration, where I titrated out, low levels of the growth factor, in the absence of growth factor, all of the control cells were dead, whereas the ones expressing BCL2 were still alive, but their numbers hadn't increased. And then as I titrated in growth factor, you can see that the control cells and the BCL2 expressing cells responded at exactly the same concentration of factor. So this meant that the cells expressing BCL2 starved of growth factor had a different phenotype to a cell that was cultured in the absence of growth factor, that was a dead cell, or a cell in the presence of growth factor that was a dividing cell. BCL2 had a completely different effect. BCL2 on its own couldn't stimulate cell proliferation. So it used to be thought that growth factor acted on a cell by binding to its receptor, and that gave a signal that caused cell survival and proliferation. In the absence of growth factor, there was no signal for survival and no signal for proliferation, and the cell would die. But these experiments showed that there must be some sort of branch and an independent regulation of cell survival and cell proliferation. So in the presence of growth factor, the cells still received a stimulation to proliferate, and if you overexpress BCL2, that had no change to the phenotype. But if you overexpress BCL2 and took away growth factor, the cells wouldn't die because BCL2 was having a specific effect on this survival pathway, but the cells also wouldn't proliferate because there was no signal from the growth factor receptors stimulating cell proliferation. So these experiments meant that there were distinct genetic control of cell division and survival. It meant that BCL2 specifically inhibits cell suicide, and BCL2 became the first component of the mechanism that regulates cell death to be recognised. And because BCL2 translocations are associated with follicular lymphoma, it meant that inhibition of cells' ability to kill themselves can lead to cancer in humans. Now, we wanted to try to make...
Later, 1 minute.
1 minute.
You're running close to time.
Right. We wanted to try to make an animal model of follicular lymphoma, and to do that, we made transgenic mice. And this shows the dot plots of the first transgenic mice we made here at WEHI. But the program for making transgenic mice was massively accelerated, and that's one of the reasons for that growth in the curve through the efforts of Sue Barth. And Sue Barth would work tirelessly, coming in at weekends to inject the mice with hormones so that she could generate transgenic mice. She generated over 25 different strains of mice, and these were able to accelerate research at WEHI dramatically. And the next acceleration came because this is the annual report from 1989. The next reason for the acceleration was the arrival of Andreas Strasser. You can see him here, looking exactly the same. I think he's still wearing the same T-shirt. But obviously it's a long time ago because his hair's different. So with that, I'll pass on the baton to Andreas Strasser.